We're continuing our studies in Chapter 20 on DNA replication and repair, and in this lesson we'll be looking at the enzyme DNA ligase and see how it functions with the lagging strand. Remember, our lagging strand contains discontinuous segments of DNA, Okazaki fragments, but we need to be able to join these together to form one continuous DNA strand. The first thing we need to do is remove that RNA primer. And that is accomplished for us by the enzyme RNase H. It's called an RNase because it digests RNA. The H comes from the fact that it represents a hybrid helix, that is, a helix of DNA and RNA. It is a 5' to 3' exonuclease, exo because it digests from the end, and it starts at the 5' end. So RNase H in our figure here is the dark green circle, template strand in blue, RNA primer in red, newly synthesized DNA in purple, and the lighter green sphere is our DNA polymerase. So RNase H will recognize that hybrid helix. This is how we ensure that it's RNA that starts to get digested. It's going to start at the 5' end and clip off one nucleotide at a time. Eventually, DNA polymerase will catch up to RNase H. DNA polymerase will essentially replace any nucleotide that RNase H clips out. In some cases, RNase H might actually remove some of the DNA within that Okazaki fragment. But again, not a problem because DNA polymerase will replace whatever RNase H cuts out. This actually helps to ensure the fidelity of replication. There's a higher error rate in those first few nucleotides that are added to the primer. And so if RNase H removes some of those, we have a better chance of making sure the sequence is correct. In some cases, DNA polymerase that's synthesizing a newer fragment may reach that older Okazaki fragment before RNase H has had a chance to remove the primer. What happens in that case? DNA polymerase has the ability to displace that primer. It recognizes that hybrid, recognizes it as RNA, and displaces it and fills in the missing DNA sequence. A flap endonuclease thereby recognizes that single-stranded flap and clips off the primer. It actually recognizes the junction between RNA and DNA. This is an endonuclease because it cuts within a sequence. Now, because RNase H might remove part of the DNA sequence as it begins its work, there's a chance that the nick between the two fragments, that is, that gap between the two fragments, might move towards the 3' direction. We call this nick translation. So we've removed our primer, we've filled in all of the missing nucleotides. There's just one more thing to do, and that is to join the fragments together. For this, we need to join a 5' phosphate on one nucleotide with the adjacent 3' OH on the adjacent nucleotide. DNA polymerase cannot do this. All it does is add nucleotides. It can't join two together that are already present. For that, we need a different enzyme. We need DNA ligase. It's going to join these discontinuous fragments. It does so by activating the 5' phosphate on the DNA strand by transferring AMP. This is very similar to the way that we activated glucose. We formed UDP glucose by transferring UMP, and that activated the molecule so that we could then add it to the growing glycogen chain. In a similar way, DNA ligase transfers AMP to the 5' phosphate, and that gives it the stored energy it needs to form that last phosphodiester bond to join those two nucleotides together. The AMP is transferred from ATP in eukaryotes and from NAD in prokaryotes. So now we have our full-length DNA strand on the lagging strand, and we've removed the RNA primers. Of course, we have one RNA primer on the leading strand, and RNase H also needs to remove that for us. In our next video lesson, we'll see a problem occurs at the ends of newly replicated linear DNA, and we'll see the steps that we take to overcome this potentially damaging problem.